Cells and organelles. So we're first going to talk about the parts of a typical animal cell. So let's get started with our plasma membrane. Our plasma membrane provides a selective permeable barrier between the intracellular and extracellular environments of the cell. So as we can see on this model, our outer portion is going to be the plasma membrane. Now there are some structures to note on the plasma membrane. So your plasma membrane is a phospholipid bilayer, so a double layer of lipids. Your phospholipids are your hydrophilic water-loving phosphate heads and two hydrophobic, meaning water-fearing, fatty acid tails. Now we can see it better on this drawing. We have our extracellular space, the outside of the cell, our cytosol, which we're going to talk about in a second, and this is the inside of the cell. Then we have our entire phospholipid bilayer with your phospholipids, your phospholipid head and your tails. Next we have our cytosol. So the main function of the cytosol is to suspend your organelles. The structure is mainly a water-based solution and it contains ions, small molecules, and macromolecules. And we can see our cytosol in this picture as the yellow kind of transparent, translucent background. Now, compared to our cytosol, our cytoplasm is everything between the plasma membrane and the nuclear envelope. So specifically, it acts to suspend the cellular organelles as well, and it contains enzymes that mediate cytosolic reactions such as glycolysis and fermentation. Now, on the practical, if I ask you to ID this structure and I only point to the yellow portion, this would be your cytosol. However, if I put an arrow or a bracket on this entire section and I say ID structure or ID feature, this would be your cytoplasm. Now moving on, organelles themselves are your little organs, and there are many types and many different functions to the organelle. The type and number of the organelles is a reflection of the cell's function. So depending upon where you are looking in the body, you might see more mitochondria, you might see more smooth endoplasmic reticulum. Organelles can be divided into two subcategories. We have our membrane-bound organelles and our non-membrane-bound organelles. We are first going to start with our membrane-bound organelles. So specifically, membrane-bound organelles, this is your biochemical activity in organelles is isolated from the cytosol and the organelles. Some examples of the membrane-bound organelles are your nucleus, your endoplasmic reticulum, both your smooth endoplasmic reticulum and your rough endoplasmic reticulum, your Golgi apparatus, lysosomes, peroxisomes, and mitochondria. The first structure that we're going to talk about is your endoplasmic reticulum. So this is a network of intracellular membrane-bound tunnels, and it can be subdivided again into two. We have our smooth endoplasmic reticulum and our rough endoplasmic reticulum. I abbreviated it here, however on the practical you may not abbreviate it, you must write out the entire name. So that would be for your smooth endoplasmic reticulum, you would have to write out the entire thing instead of SER. So let's talk about our smooth endoplasmic reticulum. Our smooth endoplasmic reticulum is called smooth because it appears smooth. With that, it doesn't have any ribosomes on the outside, as we can see here, compared to our rough endoplasmic reticulum, which we're going to talk about in a second, has ribosomes connected to it. The main functions of the smooth endoplasmic reticulum is synthesis, transport, and storage of lipids. It is also involved with detoxification of drugs, alcohol, and poisons. Now, drugs in this sense also means any type of prescribed medication. Now we can see on our model, we have our smooth endoplasmic reticulum. This is compared to our rough endoplasmic reticulum. So on this model, you can see these tiny blue little dots on the outside of the endoplasmic reticulum, meaning that this is the rough endoplasmic reticulum, compared to your smooth, which does not have those dark dots. Then on this model, we can see our rough endoplasmic reticulum here. We can't really see a smooth one. Now moving on to our rough endoplasmic reticulum. As previously stated, it's called rough because of the appearance of the ribosomes, which synthesize proteins. The main function of the rough endoplasmic reticulum is synthesize, transport, and store proteins for cells. Proteins move from the rough endoplasmic reticulum into transport vesicles, which goes to the Golgi apparatus, which we will talk about in a second as well. So again, on our model, we can see our rough endoplasmic reticulum. Rough endoplasmic reticulum. Now let's talk about our Golgi apparatus. The Golgi apparatus is a stack of flattened membranes that receive proteins from the rough endoplasmic reticulum, and then it modifies, packages, and stores them for delivery to other organelles or to the plasma membrane of the cell. So on our model, we can see our Golgi apparatus. So transportation. Let's talk about transportation a little bit more. So we talked about how the rough endoplasmic reticulum pinches off into a transport vesicle, which then goes to the Golgi apparatus. Finished proteins leave the Golgi apparatus via a Golgi vesicle. So if a vesicle becomes secretory vesicles, it releases proteins and other secretory products through the plasma membrane via exocytosis, so exiting the cell. If a vessel does not become secretory vesicle, it will become a lysosome or become a part of the plasma membrane. This is just an image breaking down the different movement through the Golgi apparatus. So again, we have our rough endoplasmic reticulum, the rough endoplasmic reticulums in the transport vesicles moving to the Golgi apparatus. Once the vessels fuse with the Golgi apparatus, it moves from each separate membrane on, and then the Golgi apparatus modifies these proteins, and then the modified proteins are then packaged in a similar way that they were in the rough endoplasmic reticulum, and then sent off into the plasma membrane, or it exits the cell through exocytosis, or it becomes a lysosome. Here's a video describing the protein transfer from organelles more in depth. I suggest starting at second 57. Now let's talk about our lysosomes. So lysosomes are membrane-enclosed sacs that contain digestive enzymes. The main function is the breakdown of intracellular debris. So on our model, we can see our lysosomes here and here. Now, similar to lysosomes, we have peroxisomes. These are membrane-enclosed sacs that contain catalysts and other oxidative enzymes. The enzymes break down lipids and toxic substances by first converting them into hydrogen peroxide and then breaking down the hydrogen peroxide into water and oxygen. So on our model, the yellow would be our peroxisomes. Next, we have our mitochondria. So mitochondria is usually referred to as the powerhouse of the cell. These organelles are the site of cellular respiration, so the metabolic pathway that utilizes oxygen in the breakdown of food molecules to produce ATP. So on our models, we see our mitochondria. And on this model, this is as well our mitochondria, however, these are cross-sections, so they look a little bit different. Now let's talk about the nucleus and nuclear membrane. So the nucleus is the center and it contains the cell's genetic material, also known as your DNA or your deoxyribonucleic acids. Your nuclear envelope is going to be the outer membrane of the nucleus, and it also has nuclear pores, which many students often confuse with ribosomes. So let's look at our model a little bit. So if I put a circle around this entire structure and said ID, organelle, you would say nucleus. However, if I pointed only to the blue portion and I said ID structure, outer portion, or be specific, this would be your nuclear envelope or your nuclear membrane. Then we have our pores. So these dark blue dots, if I pointed to these structures and said ID structures and said pointing or hint to the dark dots, you would then know that these are your nuclear pores. Now within our nucleus, we have our nucleolus or our nucleoli. Our nucleoli synthesize our RNA and resemble ribosomes within the nucleus. So it's usually a darker staining body within the nucleus, and it sometimes resembles that ribosome. So let's look, we have our nucleolus and our nucleolus. Now let's talk about our non-membrane bound organelles. So our non-membrane bound organelles are any organelles that are in direct contact with the cytosol. And some examples to remember are your ribosomes, your cytoskeleton, and your centrosomes and centrioles. So let's talk about our ribosomes. So our ribosomes are the sites of protein synthesis, and they may be bound to the ER, so these are your fixed ribosomes. If you find ribosomes that are just kind of floating in the cytoplasm, these are going to be your free ribosomes. So let's look at this model. So anything that is attached to your rough endoplasmic reticulum is going to be your fixed ribosomes. And then we can see here are free ribosomes. And on the practical, I want you to be very specific. If I point to these and say ID organelle and you only put ribosomes, it will be wrong. You must put free ribosomes. Now let's talk about our cytoskeleton. So this provides the main structural support for the cells and is composed of microtubules, intermediate filaments, and microfilaments. So our microfilaments, they maintain and change the cell shape. They participate in muscle contraction and cell division. Our intermediate filament, again, they provide structural support, and then they stabilize connection between the cells. And last are microtubules. So they radiate from the centrosome. Again, they maintain cell shape. They direct movement of organelles and DNA, and they allow cell motility, so such as your cilia and flagella. Now we have our centrosomes and our centrioles. Our centrioles are microtubules, and our centrosomes are a pair of centrioles at right angles to each other. So if I were to put an arrow on one of these, or if I were only to circle one and say ID structure or ID organelle, you would say centriole. If I put a circle around both, you would say centrosome. Again, a circle around both, centrosome. If I only pointed to one, centriole. Again, circle around both, centrosome, only pointing to one, centriole. Now we have our cilia and flagella. So these are projections of the cell containing cytoplasm and microtubules capable of movement. So we can see in this image, we have our cilia, which are grouped on cells that move objects across their surface. Next we have flagella. So these are going to be longer and they usually are singular to propel a cell. So think of sperm. Again, we have our cilia, close-up picture, and then we have our flagellum on a sperm. Now we have microvilli. Microvilli are extensions of cells and they are not capable of motion. They are very similar to cilia, however they are very small, and the main function of them is to increase the surface area to increase the absorption of food. So again, we have our cilia, and our smaller cilia are actually microvilli. The next portion that we are going to talk about is cell division. So first we're going to talk about the cell cycle. This is a series of events that cells go through as they divide. So the main function is cell growth, preparing for the division, and then divides to form two daughter cells. There are two main phases of the cell cycle. We have interphase and mitosis. Interphase is the growth that occurs between cell division, and most of the cell spends its entire life in this, so 90% of the cell cycle is actually spent in interphase. Interphase can be divided into three main phases. We have G1 phase, S phase, and G2 phase. G1 phase is the first phase where the cell grows and matures. S phase is the DNA replication. G2 phase is the last phase where the cell nucleus prepares for division. And we can see on this model, if I said ID phase, you would say interphase. Now let's talk about mitosis. So mitosis is a type of cell division, and the cell usually spends about 10% of its cell cycle or cell life in mitosis. The result of mitosis is two daughter cells, each having the same number and kind of chromosomes as the parent nucleus. It's typical of ordinary tissue growth and repair. So just remember PMAT and C. So you have prophase, metaphase, anaphase, telophase. You also have cytokinesis. Some people consider cytokinesis as a part of telophase. Some people consider it as its own phase. All right, let's talk about prophase. So prophase, this is your chromatin condensing, and it shortens and thickens into chromosomes. So we can see our chromatin condensing into our chromosomes. Our spindle fibers begin to form, and then our centrioles separate and organize into spindle fibers. The nuclear envelope begins to break down. We can see these little ridges. Next we have metaphase. So when I think of metaphase, I always think of middle, because our spindle lines up the sister chromatids along the middle of the cell. This is also known as your metaphase plate, the center. Each chromosome is connected to a spindle fiber at its centrosome. Then we have anaphase. Anaphase is essentially when the chromatids are pulled apart. So the spindles shorten, pulling sister chromatids apart towards the centrioles, creating individual chromosomes. Then we have telophase. So the chromosomes gather at each pole at opposite ends of the cell, and the chromosomes start to lose their distinct shapes. The nuclear envelope then begins to reform. We can't see that really in this model, but just know that is one of the features of telophase. Now in this is actually a little bit of the last part of telophase, the beginning of cytokinesis. So right here we have our nuclear envelope beginning to reform. We still see those ridges. We see our chromosomes starting to lose their distinct shape, yet we still see our spindle fibers. However, with our cytokinesis, a cleavage furrow begins to separate the daughter cells. So you can see this cleavage beginning to form to separate these two daughter cells. Each daughter cell has an identical set of duplicate chromosomes. And finally, the last part of cytokinesis, that full cleavage furrow is beginning to form and starting to split off these cells. I highly suggest watching this mitosis in real time. It's a very interesting video. And that's it for our mitosis. These are the different phases of the cell cycle for mitosis.